most common cause of Stargardt's disease is a mutation of the ABCA4 gene. How does this mutation affect the retina?
The ABCA4 gene is very important for the function of our central vision, which we call essentially the foveal vision or the high resolution vision. So ABCA4 is a protein that somehow gets rid of toxic metabolites. And if the gene is malfunctional, basically these metabolites would accumulate in the retina, which leads to cell loss. And central photoreceptor loss essentially leads to loss of the high resolution vision, which will essentially mean patients lose the ability to read or recognize faces.
Why is Stargardt's disease particularly suitable for testing gene therapy approaches?
So in general, any disease that can be attributed to a single gene, like in this case, an ABCA4 mutation, in general is a good candidate for gene therapy because we know exactly what the problem is. Now, the difficulty with ABCA4 is that it's a relatively large gene. Therefore, if we want to just perform a classic gene replacement therapy, current technology does not really allow to do that. So on one hand, it's ideal because we know it can be potentially treated by gene therapy. On the other hand, we need new technologies such as base editing, which we'll be talking about today, to really tackle the gene therapy challenge for ABCA4.
At the IOB, you work with this base editing technology. Can you explain how this specific method works and what advantages it offers?
Yes. So base editing is a gene correction approach. The approach is based on this CRISPR-Cas9 technology, which was awarded the Nobel Prize to Jennifer Doudna and Emmanuel Charpentier. And essentially, base editing is a more sophisticated version of this CRISPR-Cas9 gene editing method. The idea is that the human genome consists of 3.2 billion letters or bases. And if one of them is, if there's a typo in only one of them, this essentially could lead to blindness. And base editing makes it possible for the first time to really correct the mutation and only the mutation that affects the patient. Essentially, imagine you have to find a single letter out of 3.2 billion letters. So this is what base editing does. It finds this typo in the text and makes a correction back to the wild type sequence. Now, once this correction has been made, because it's at the DNA level, this is once for a lifetime effect, as long as the cells are still present, which we hope that after this editing event, we will be able to rescue the cells for a lifetime.
What were the biggest challenges in developing this gene therapy?
When we started using this new therapy, it was demonstrated that this works very well in cell lines, so cell cultures, or in the mouse retina. But we wanted to make a therapy, a human therapy, using this technology. So it took several years, 5-6 years, to really adapt this technology from cell lines all the way to the human retina. So for this, we used uniquely human models. In IOB, this is a very important model system. And initially, we just observed that only a few percentage of the cells could be corrected. And after several years of optimization, we're right now at the stage when we can correct 80% of the affected cells. So that means we can correct 4 out of 5 disease cells. So I think that was the difficulty to how you bring a technology, an immature technology, from a petri dish to a human model system.
Could you say something about the models you work with at the IOB?
Our main philosophy is that if you would like to make human therapies, you have to develop in human models. And we are still using, of course, other models, but for us, these human models are the most important. And there are 2 models that come into my mind. The first one is human retinal organoids. We can essentially take a skin cell from a patient and turn it into a retina-like structure in a dish. And we can make thousands of this. And essentially, this structure is called an organoid. This will have the mutation, and this allows testing of a new therapeutic approach. The second option is human retinal tissue, which was developed by Arnold Szabo at Semmelweis University. Here we have the option to study the human retina from multi-organ donors, essentially as it is, and we can try to see if I can perform a base editing directly in the human tissue. Now, this human tissue is from non-Stargardt patients, but with base editing, we can figure out ways to assess therapeutic efficacy.
What were the key findings of your studies?
So the most important is that we have been able to demonstrate that we can perform the correct editing event, so the correct correction of this mutation in 4 out of 5 cells. We think if one would correct in 1 out of 5 cells, this would already lead to therapeutic efficacy. So we're quite confident if the same thing would be replicated in a human who is suffering from Stargardt disease, this would be at the therapeutic level. The second important finding is that we have seen an incredible precision. So we have not seen any other off-target effects. That means we have not seen any other editing event other than the Stargardt disease causing mutation, which is a remarkable finding if you think about these 3.2 billion possibilities of changes that one can induce if the therapy is not good. So the efficacy and the precision together, I think this is a unique constellation which makes me excited about this therapeutic approach.
What further steps are you planning to take to validate the safety and efficacy of the therapy and prepare it for clinical trials?
I told you that this technology already reached an efficacy, what we think is therapeutic, but further steps are necessary. So first of all, we have to make sure that we use as low dose of this gene therapy as possible, because at high dose, people have seen toxicity with these vectors. So we have to make sure that our vector is just as optimal as possible in order to reach high editing levels from as little input gene therapy agent as possible. But this is something that we are actively working on. And it is also true that if one would like to conduct a clinical study with Stargardt patients, we need regulatory approval, and we have to perform certain tests for the safety of the approach, which are essentially mandated by regulatory authorities, which are very standard tests, and simply they have to be performed according to regulations.
Which vectors did you work with?
So we are using vectors called AAV vectors, or adeno-associated viral vectors. So AAV vectors have already received clinical approval for other diseases, including a rare eye disease called Leber's congenital amaurosis type 2. This is the Luxturna therapy. This is the same vector and the same procedure as the Luxturna procedure. So it's a subretinal injection of the vector. So AAVs are also approved for other diseases such as spinal muscular atrophy. So in terms of the vector system, this is not new. What is new is that we're delivering this base editing cargo, which essentially makes this precise change.
Could similar base editing approaches be adapted for other inherited retinal diseases?
Absolutely. So I think it's important to mention that what we developed here is specific for one mutation. Now, there are many other mutations that can cause Stargardt disease, but there are many other mutations that can cause other diseases. As long as the mutation affects a single base, and as long as this base change is either a G to A change, so guanine to adenine, or a thymine to cytosine, we can make the reversal. So the reversal is from an adenine back to the G, or a cytidine back to a T. these mutations are called base editable mutations. Actually, it turns out that the majority of the patients in Stargardt disease, 95% of the patients have a base editable mutation. So people have two mutations. And according to our calculations, that in 95% of the cases, at least one of them is base editable. But it's important to say that you would need a different targeting system. You need the same base editor, but a different small sequence that targets the base editor, not to this mutation, but to another mutation. But of course, the technology holds a great promise because it's not only applicable in Stargardt disease, but other diseases which are inherently caused by base editable mutations.
Which partners is the IOB working with on this research project?
So the project was funded by the Swiss National Foundation. So I have an academic grant for this. And this had a tremendous help, first of all, for us to get started to develop the models where we could start testing the therapy. Then IOB, in a subsequent step, partnered with Beam Therapeutics, a Cambridge, Massachusetts-based company that has the exclusive license for the base editing technology. So we had a great collaboration with them. We worked for several years on developing this technology further. But again, academic funding is very important because the last part of the project was again funded by the Swiss National Foundation. So essentially, IUB is an academic research institution where academic funding plays a critical role. While, of course, because we're talking about therapy development, we have to identify industry partners to bring therapies closer to patients.
What makes the IOB one of the leading centers for ophthalmological research?
I think there are two main points I would like to mention here. So one is that in IOB, clinicians and researchers really work together in order to bring therapies closer to the clinic. So IOB has really ambitious goals to develop a therapy, for example, for Stargardt disease, or to develop a vision restoration approach. That's another therapy we are working on. But in order to tackle these really big goals, we need complementary expertise. We essentially need people who are expert in molecular biology, people who are experts in organoid technology. We need engineers, we need data scientists, but we also need medical doctors, clinicians who see these patients. And I think this chain of events have been built in a way that this functions coherently to tackle really complicated tasks, such as, for example, developing base editing for Stargardt disease. The second main point, which makes IOB really unique, is the application of human models. Because, again, several therapies before have been developed in, for example, in mouse models. And unfortunately, often these mouse models do not translate to human patients that easily. So we have a different philosophy because when we say we're going to develop a new therapy, the majority of the development happens in human models. Of course, one can start to validate in non-human models as well, for example, in mouse models. But for us, doing uniquely human models in IOB is of critical importance, which places this institute in a very unique position.
What long-term visions do you have for the use of gene therapies in ophthalmology, and how could this change the care of patients with rare retinal diseases in the next 10 years?
So I think in general, gene therapy is still in early days. We think gene therapy has been around for a long time, and that's true, but we still don't understand many things around the application of vectors, for example, in the human eye. And the tools that we have today are much more sophisticated compared to the tools that we had several years ago. So the gene therapies that are currently in clinical development are based on a technology that existed 10 or 20 years ago. So I think this is a natural evolution in the field that is just really exciting, that what we have now, I really hope that it will have a more transformative change to patients, because I am not satisfied so far with the progress of gene therapy. But I think this is just because we understand still our understanding is still limited around these gene therapy technologies. But I think gene therapy has a clear way forward as a therapy, because especially this therapy, because it acts at the root cause. So I think with base editing, especially, this is the best we can do. So if we can correct the mutation in the cell that is affected by the disease, it's hard to think what else we could do. And in my opinion, this is an excellent way forward. The question is, how do we do it? Which tools do we use? And when I say tools, I talk about the molecular machinery, the virus vector, the components of it. So these are molecular tools that this will only work if all these components are assembled in a way that that is correct, and it will lead to a safe and effective application of the therapy. I am in general very hopeful for gene therapies, especially in the eye, because the eye is rather a small compartment with a relative immune privilege where we can really administer these gene therapy medications. But we have to acknowledge that the field is just more complicated that what we have hoped for. And importantly, we have to really acknowledge that whatever we find in mice might not work in human. Therefore, we have to establish efficacy in humans before going to patients. And that's one of our main missions here in IOB.